Good morning. I'm Cam McDougall. I'm the Executive Medical Director for Neurosurgery and co-director of the Swedish Neuroscience Institute. It is my pleasure to be here this morning with Dr. Charles Cobbs for another in our John Jane Continuing Lectureship Series. Charles is an old and dear friend, known to me since his days as a resident at UCSF. Charles was mentored under the legendary Charles Wilson, and while in his residency there did a molecular oncology fellowship, and when he finished his residency moved to the University of Alabama, where he was an associate professor for a number of years before returning to San Francisco at CPMC, and he has continued his research throughout this time in molecular oncology. He was involved in identifying CMV virus as an important oncogene contributor and has diligently pursued this for his entire career. He joined the Ivy Center in 2013, and he's been the driving force and sustainer of the Ivy Center since that time. His work continues today in effective collaborations with ISB. He is involved in R01 and R21 grants, continuing his research into the pathogenesis of glioblastoma. And we are grateful for his ongoing work and delighted to have him here this morning to talk to us about immunotherapy for glioblastomas. Charles, please. Thanks.
Thank you. Thank you very much, Cameron. It's fun to be able to work with you after all these years still. So I have a talk about making a cold tumor hot, and the cold part you can hardly see, but that's kind of a metaphor for what's going on in glioblastoma. The immune system can't see it. I'm really excited actually to start the talk with a slide here to describe a patient of ours that I operated on about 3 weeks ago who's going to be our first patient to enroll on a novel clinical trial. And I think it's appropriate to mention this because it's a full circle for me for a concept that was a crazy idea and is now being used for hopefully helping patients with recurrent glioblastoma. So VBI Vaccines is a vaccine company, infectious disease vaccine company, but also interested in cancer, based out of Cambridge, Massachusetts. And I spoke to the researchers there years and years ago about a concept for CMV vaccine against glioblastoma. In September, they announced a multi-center randomized controlled study of phase 2B, which had fast-track designation and orphan drug designation based on some preliminary work in the phase 1 component. This VBI 1901 with GM-CSF, which is a adjuvant at the site of the vaccine injection, has a 44% disease control rate from the first 16 patients with recurrent glioblastoma, which is pretty striking. The thing that I want to point out is the vaccine is composed of two viral-like particles coated with a GB protein and a PP65 protein. What are those proteins? Those are proteins you're going to hear a lot about in a minute because they are CMV virus proteins. So I'm going to start the talk about the first 15 minutes talking about CMV and how it came to be a concept in glioblastoma and how it may inform us and contribute, hopefully in the future, to immunotherapy for glioblastoma. So years ago when I was first at UAB, I had a patient who looked me in the eye and said, "What causes glioblastoma?" after they had been diagnosed. And I had to give the sort of pathetic pat answer that we often give is like, oh, well, we don't know, but there are some genes associated like EGFR and mutations and blah, blah, blah. And then I had been reading a book by a hero of mine, Richard Feynman, who is, as you know, a physicist who would ask deep questions and kind of push people to dig deeper. And so I started really thinking about this and realized that when I was at UCSF, for instance, I hardly ever saw lower socioeconomic GBM patients, although I saw those patients for all other disorders. And then I started looking into this, and there was weird stuff about the epidemiology, and that panned out, and that's a real finding. There are other weird things about GBM, such as birth order being relative to your risk for GBM. If you're the only child or first or top in your family in the sibling order, you have a higher risk. So what does that have to do with anything? Well, it turns out those are also predictors of infectious-related complications from some herpes virus infections like Epstein-Barr virus. And so sort of asking around other mentors of mine if they knew anything about what caused glioblastoma, I realized that we really don't know, and no one really has a good answer. So I thought I would start doing some deep thinking and trying to see if there's any possibility that there could be something we haven't, a stone that has not been turned over. So I went back to kind of the basic concepts. At the time, I had been in a lab previously at UCSF, and we had discovered that nitric oxide, a free radical, was present in glioblastomas and might drive some of the processes. And it's a free radical associated with inflammation. And so, if you look at how cancer arises, most people think of genetics in cancer. Well, genetics doesn't really cause cancer. Cancers start and then have mutations, which is a different concept. Most cancers start with epithelial cells, and glioblastomas are neuroepithelial. Most of the time it's due to an injury with oncogenic events, like in your skin cells getting mutated with UV radiation. But in a lot of cases, 20% of cancers in humans that we know of, pathogens get into the tumors, into the cells, and chronically infect those cells. Some of these pathogens promote mutations, and those mutations over time can cause these cells to transform into tumor cells. When the original pathogen comes in, you get a huge elicitation of the innate immune system, and if you don't remember, those are macrophages, dendritic cells, and monocytes, and NK cells. They usually come in and blow away the pathogen. But what happens in this concept of pathogens and cancer, which is, this is sort of the ongoing concept here, is that in the initial stages, the pathogen may come in and you get an innate immune response. Monocytes, macrophages, and NK cells will blow up the cells, send bits and pieces to dendritic cells, which would go to your lymph nodes, and tell B cells and T cells to come back and eliminate. That's the adaptive response, and usually the pathogen is gone. However, if the pathogen gets into those cells and evolves in a way that it stays under the radar of the immune system and causes more DNA damage or mutations, then over a while, you have an equilibrium, so the pathogen persists, and this can go on for decades, like in the cases of HPV and cervical cancer. And then at some point, the pathogen may cause so many changes, like in HPV, that can mutate, it can alter P53 and RB, and that will lead to cancer. And the immune system at that point has essentially become tolerant and unable to control the pathogen. So I started thinking, could a pathogen be associated with GBM? And it was kind of a crazy concept or thought, because I talked to some leaders in the field, and they said, oh, of course, somebody would have found it by now. But there's a difference between looking and really looking really hard. So I thought about this and thought, it's a crazy idea, but I'll have a person in my lab. We'll just start going through a list of pathogens that could be associated with an infection in the brain and potentially lead to cancer. And I said, if I wanted to do this, who would I be? I would be something that causes persistent infection. I would want to infect stem cells in the brain. I would want to cause DNA mutations. I would want to make an immunosuppressive environment, and I would want to promote the properties of GBM in general. It turns out that just a few minutes on PubMed led me to CMV as a top candidate. It's the unknown public health crisis that you never think of. 1 out of 200 infants are born with CMV congenital infection. Most of those don't have outward manifestations, but it is the #1 cause of birth defects and mental disability in humans, more than Down syndrome or anything. This virus has a predilection for the brain subependymal cells. Here's a picture of a 1-year-old brain with destruction of the subependymal zone area, an infant that has microcephaly. And what you're going to hear about more today is proteins that are associated with this. The virus encodes for IE1 gene, which is highly mutagenic and promotes cancer pathways. The PP65 tegument protein, which has a potent anti-immune function, and the surface glycoprotein B, which has the ability to cause cells to fuse together, leading to mutations in cancer. But this was not considered a cancer virus. And one thing I'd like to point out is if a virus like this were to get into stem cells in the brain, it could wreak havoc, going back to the original concept of persistence and immune evasion and escape. If a cell divides and you have a daughter cell and that cell has acquired mutations, that cell is mitotic. It's not going to divide anymore. However, stem cells divide asymmetrically, so the original cell that's infected, if it acquires more and more mutations, it's going to have a higher and higher risk of causing changes that can lead to cancer. So I was thinking about this, and then a paper came out from an obscure journal, not really obscure journal, but from an obscure group that studied CMV in mice, which is highly correlated with humans in many ways. So in mice, they created a virus that turns blue when it's activated. It turns out if you infect mice with mouse CMV, they do fine. You don't see the virus anywhere. They just have CMV infection, like 83% of adult humans. They live the rest of their life with no problem. But if you take their brains and slice them and put them in culture in the absence of an immune system and they grow for a few days, the virus will get reactivated. Where does it reactivate? It reactivates in the subependymal zone. And the cells that are reactivated are positive for Musashi and Nestin, which are markers for stem cells. So the concept suggested that many adults may be walking around with a CMV infection in the subependymal zone of their brain without knowing it, and these cells could be persistently infected, and we think those are the cells that start GBM. So I had this person in my lab, Louis Harkins, start doing some immunostaining for various viruses, and the first studies we did on CMV were, I looked at it and I said, okay, that was a dumb idea, they're negative. And she said, no, they're not negative, they're just very low levels. So we spent months and months developing extremely sensitive techniques to detect immunostaining of CMV antigens, and we found that most GBMs express PP65 and IE1. And we corroborated this with RNA and DNA in situ hybridization. So the first thing that happened after that paper came out was that there were crickets. Nobody said anything. And then we started getting the same papers that said this is not repeatable. We can't reproduce your data. And as a surgeon, I took that as a compliment. But in the scientific community, that is really the kiss of death. So I kind of we kind of went dark and just started focusing on the possibility this virus could actually contribute to the pathogenesis of GBM. So we knew signaling pathways that activate downstream molecules that drive GBM, and we started looking with different experiments to see if IE1, these other viral proteins, could drive these pathways, and we published multiple papers on that. One of the first events that's thought to activate gliomogenesis was highlighted by one of my colleagues at UCSF, Arturo Alvarez Bula, where they just showed that PDGF, if expressed in the subependymal zone and the CSF, could activate a certain type of stem cell which has a PDGFR alpha receptor. And just doing that with the growth factor could cause small gliomas to start. They weren't mutated, they didn't have genetic mutations yet, but they were highly prone to resulting in GBM. So we started looking at these signaling pathways, and we found that CMV, just by adding it to cells, fibroblasts or tumor cells, could activate all these pathways. And strikingly, of all the receptor tyrosine kinases that can get activated, this is a panel with 42 different ones. Only PDGFR alpha was activated. And we showed that activation of this growth factor receptor was critical for CMV to infect the cell, and it was a novel receptor for the virus to actually enter the cell. We published that in Nature because it was considered for the virology community important as well. But what was really exciting is that just adding the virus to the tumor made the tumor grow as if you were adding the growth factor PDGF. We went on to show that that was due to glycoprotein B on the viral cell surface, and then when we did Western blots, you can see a bunch of GBMs here. Most of the tumors had glycoprotein B in them. Many of them also expressed PDGFR alpha, and we showed that that can be a driver in an autocrine and paracrine way of tumor growth. If we put the viral protein into tumor cells, they grew like crazy, and these tumors grew very aggressively, and they would literally metastasize up and down the spinal column. The next thing we focused on, because people were having trouble detecting the virus, was the fact that we felt that the virus in these tumors, sort of along with the equilibrium and escape concept, kind of go underground and seem to infect only a subset of the tumor cells, although the proteins can be found throughout the tumor, and those were the stem cells in the tumor. So we started isolating glioblastoma stem cells right from patients, which are CD133 positive, so we could sort the cells, and we found that while 99.5% of the tumor cells may be negative, the stem cells were the ones that seemed to retain the virus. And then we asked the reverse question: do they drive the stem cells of the tumor? And here you can see experiments we did where we grew the tumor spheres in stem cell conditions so that they're stem cells, and we co-stained them for viral proteins right out of patients. So they're positive for the IE1 protein, and this correlates with nestin, which is a glial stem cell marker. Strikingly, you can see here, this is a tumor sphere positive for the IE1 protein. If we knocked out the viral protein IE1, the protein goes away, as you see here, but also the tumor spheres stopped growing, and we could show that that was associated with a over 50% knockdown of all of these critical stem cell biology markers. So we had evidence that the virus can initiate the tumor growth, can promote the stem cells, and then we collaborated with a group at Harvard in Nino Chioka's lab where they had mouse models. And they showed that if you infect a mouse with CMV at birth, just like a human, for instance, and then you put a tumor into their head later on, compared to a mouse that never had CMV infection, then those tumors will grow dramatically more rapidly than the ones without CMV. Here, that compared to that. And that was associated with massive recruitment of parasites and angiogenesis. Also, very interestingly, if you treated those two mice with cidofovir, an antiviral drug, you could prevent that tumor growth. So that suggested there was a therapeutic angle. So all this was going on, and there were so many people that couldn't reproduce a lot of findings that it was even highlighted in a two-page article in Science, unfortunately with me as the poster child for controversy, which sounds kind of exciting, except that when you're applying for R01 grants from the NIH, if you're considered controversial, it's the kiss of death. So we had a lot of trouble, and we lost our R01 funding about 8 years ago because of from some of the comments, the fact that a lot of people thought that this was not repeatable. Nevertheless, that led us to pivot into another area of immunotherapy research, which, as I'll show you in a few minutes, turned out to be a blessing in disguise. In the meantime, some of my colleagues became very interested in this concept of CMV and glioblastoma, not from the perspective of, is it driving the tumor, is it causing the tumor? To really prove causality in any disease is almost impossible. But John Sampson at Duke said to me, he's like, I don't care what this thing is doing. If it's producing viral proteins, I can target that with an immunotherapy. So the Duke group and another group in Australia at Queensland Hospital and some other people started looking into the concept of if a patient has a GBM and you show that there's PP65 antigen, which is the dominant antigen, could you take the gene for that, put it in their own dendritic cells, and those dendritic cells could be given back subcutaneously with an adjuvant, and that adjuvant would allow those dendritic cells to go to the local inguinal lymph node, in this case, and those lymph nodes would allow the dendritic cells to train T cells to attack CMV PP65. So the concept was give the patient dendritic cells with PP65 activation, or give them dendritic cells and co-culture their T cells, make a pool of T cells that are now really active against CMV, PP65, and then give them back to the patient. I sort of thought that didn't sound like it was likely to succeed because it's just one viral protein, and how do we even know those tumor T cells will get into the tumor? But Dwayne Mitchell at Duke at the time started doing experiments with patient-derived GBM cells and T cells trained for CMV, and he showed those T cells could specifically kill glioblastoma cells, but if they were trained against PP65 viral antigen, but not if they were trained against other things like flu or survivin. So that was almost unequivocal evidence that the immune system could attack a GBM cell based on a viral protein. And to make a long story short, they published 2 phase 1 studies. The first was published in Nature, the second in Clinical Cancer Research, where they did this strategy by vaccinating with the dendritic cells with GM-CSF as an adjuvant or tetanus toxin. And small numbers of patients, however, of the patients that received the vaccine with the adjuvant with dendritic cells against PP65, in these phase 1 studies, the median survival was almost 4 years for GBM, which is striking compared to traditional survival. And it seemed to be related in analytical studies to the immune response to CMV. For instance, if you didn't have an immune response, or if the dendritic cells didn't get to the lymph nodes, there was less of an effect. So it kind of added up. The next few slides are just all the studies that have occurred in the last few years that have been published or else are ongoing now studying this. I didn't mention that one of my colleagues in Stockholm at the Karolinska started treating patients with valganciclovir, and she published a letter in the New England Journal, which was not prospective randomized. Nevertheless, she had striking data that if you treated GBM patients with valganciclovir, the overall 2-year survival was 70%, and overall 4-year survival was 27%. Now, that was contested hotly by neuro-oncologists, showing that it was a biased study, but she and others have gone on to show with antiviral drugs, oral valganciclovir, which is basically non-toxic antiviral drug, that there is evidence it can improve survival. And so these are published studies. There are several published studies that I just mentioned, one published in Nature in Clinical Cancer Research that showed 4 patients without progression and really long overall survival. There are a bunch of other studies, including my colleague in Australia that I mentioned, who had been giving T cells that were activated against PP65 from patients, giving their own T cells back that had been revved up to attack this virus. And they gave in 2 small studies to recurrent GBM patients. And some of these patients, I talked to Rajiv recently, some of these patients are out past 10 years now with recurrent GBM. So it seems like there are patients who are having a really significant impact from this therapy. And I'm not saying that this is the end of the story, but it's just the beginning. But these are ongoing studies now. No, these are published studies. And these are all of the ongoing clinical trials now with immunotherapy against CMV. So there are at least 10 trials now, including the one that I mentioned on the first slide that our patient is involved in. So what's going on here? So briefly, I mentioned Dwayne Mitchell sort of hypothesized that if you look at a tumor and you think that the ones that are red here are the ones that are infected with CMV, if you activate T cells specific to CMV peptide or CMV antigen, you can either kill these guys and the other ones will survive. Maybe you can kill these guys and you'll actually cause more cells to come in from the outside that will come in and say, hey, there's a viral infection, and that will continue to chomp away at the tumor and the tumor cells that are infected. And then this paradigm here is one that we're going to talk about most, which is if you can get antiviral killing with T cells, what happens is you will blow up these cells. They will release viral antigens. They will also release antigens of tumor cells in a stressful environment. Your innate immune system, which I'll talk about a lot further in a minute, has the ability to detect these damaged molecules, and that can activate that innate immune system function that I mentioned in one of the early slides about activation, equilibrium, and escape. What's thought now is that if you activate these dendritic cells, they can come back and they can have cross-epitope spreading. So now they may be activated against a viral pathogen, but they may come back, and in this re-teaching of these dendritic cells, they may also suddenly see that the tumor is a foreign body too. So you get cross-epitope spreading, so-called. So I'm going to finish this sort of part of the talk with this paper that was published from a group in Norway. They looked at glioblastomas in their patients. They detected PP65 and IE1 proteins in a bunch of patients. They found that most patients with GBMs were seropositive versus controls to CMV. But the most important part here, and I couldn't for some reason highlight this part, the T cells that were specific to CMV in the tumor were dramatically coated with two proteins we're going to talk about, CTLA4 and PD1, which are checkpoint proteins that tell the T cells do not function, don't do anything. So these are the bane of immunotherapy now, and I'm going to switch gears in the talk and start talking about immunotherapy in general. So the T cells were there, but they're not functioning. So maybe that's why this is not working. So the concept I'm going to talk about now is, having worked with brilliant people at the Institute for Systems Biology, like Leroy Hood, I want to see if we can go back and leverage a systems approach to reactivate this immune function. So if you look at the cancers that have been breakthroughs for immunotherapy, melanoma, head and neck, we know that those tumors respond well to checkpoint inhibitors. Why? Normal cells, have expression of proteins that are produced, and your T cells have been trained not to attack them. However, in a tumor like melanoma, with thousands and thousands of DNA mutations, these eventually will encode for RNA mutations and novel proteins, and those novel proteins will be presented at the surface of the cell. During the equilibrium and escape, the T cells that are supposed to kill those new antigens will become coated with these checkpoint molecules and will not function. However, the Nobel Prize was awarded a few years ago for the breakthrough from Jim Allison and others who discovered these drugs that can block these checkpoints, releasing these T cells to activate and kill these tumor cells. That's how these drugs work. So it's sort of a paradox. The more mutations you have, the more likely you will respond to a checkpoint inhibitor drug. And this is a paper from Nature that illustrates that melanoma and squamous lung cancer have, on a log scale, hundreds, if not thousands, more mutations in general than a lot of tumors like glioblastoma. So on a stochastic level, these tumors are going to be more sensitive to checkpoint inhibitor drugs because they have more neoantigens on the surface. Glioblastoma doesn't have that many neoantigens. We've tried to attack glioblastoma by focusing on a couple of molecules like EGFRV3 or the IL-13 receptor, but one or two little things are not going to knock out a GBM. I'm here to tell you, and that's what the science shows. So if we took a holistic view, could we take this escape environment for GBM, shove it backwards, and do a massive reboot with elimination? And that's what I'm here to talk about. This is a good paper. It's a review of innate immunity, and I've really done a deep dive on basic immunology recently. Innate immunity is, as mentioned before, dendritic cells, NK cells, macrophages. If you have a viral infection or some damage to the cell, these guys come in, they just kill anything in sight, blow it up, and they hand off little bits and pieces to the dendritic cells, which go back. And in this environment, you get a cytokine storm. So you get a storm of activated molecules. These guys, dendritic cells, take these bits and pieces to the lymph node. They tell the T cells, go find this thing and kill it. And then you have that second peak of the adaptive immune system where B cells produce antibodies and T cells produce cytotoxic T cells, and they come back. So these are the things that are required to get this thing rolling, which is pathogen and damage-associated molecular patterns and tumor-associated antigens. I just told you we don't have a lot of tumor-associated antigens that we know of in glioblastoma. And thinking about this and talking to some experts, I realized that we can't solve this problem the way we're doing it now, which is to make one molecule, let's say CMV PP65. We can't make a vaccine to one molecule and expect to wipe out a GBM. My bias is that we need to rethink the whole thing. We need to go back to square one and do a holistic approach with an innate and adaptive immune massive response, which I like to kind of call a four-alarm fire. What's a four-alarm fire? A four-alarm fire is when you have a fire so big you have to call in fire stations from 4 different places. What I want to do is turn a glioblastoma into a raging fire of immune activation and a cytokine storm that will send signals around the body to have more adaptive and innate immune cells come in, to have antigens that are present perhaps from pathogens that will cause epitope spreading, and then have a massive holistic wipeout of the tumor. So how are we going to do that? So I just told you the way to reboot the innate immune system is with pathogen alarm molecules, damage alarm, tumor-associated antigens that we don't have yet, and then reactivating the innate and adaptive T-cell responses. So what I'm going to tell you next is something that most people have never heard of, but it's really, really exciting. So 6 years ago, there was a paper in Nature that I read about T-cell receptors in different tumors by Evan Newell's group. He was in Taiwan. I read the paper, and it was really interesting because he said, here we show that lung and colon cancers have T-cells specific for tumor antigens, but also some epitopes for viruses. And here's an example. PP65 and IE1 T-cell receptors are present in these tumors. So I actually called him, and he said, hey, I'm actually moving to Seattle to go to the Hutch. And I said, so why are these T-cells in these tumors? Is it because they're infected? He's like, I have no idea. So that was a dead end. So I sort of put that in the memory bank, and then a paper came out 3 years ago. This is from David Massapust's lab at the University of Minnesota. I believe he's probably one of the most brilliant people I've ever spoken to. He read that paper, and he said, these T cells are specific and poised to go after these very specific antigens of these pathogens. So he decided to take tumors in mice and coat the tumors with bits and pieces of these viral antigens. And he was able to reactivate an immune response that wiped out the tumor because the T cells came into the tumor, wiped out the tumor because of the viral antigens, and then had epitope spreading and wiped out the tumor. So he said the peptide theory described in this study operates by tricking antiviral T cells into perceiving a local reinfection that broadly activates innate and adaptive immunity. So it's taking that elimination, equilibrium, and escape model and going back to square one and rebooting. And then another paper came out 2 years ago by John Schiller's lab at the NCI. He's a famous guy at the NCI. He's the guy, along with Doug Lowry, who basically put the HP vaccine out there. He's a cancer immunologist, and his lab, for whatever reason, started honing in on CMV. And they found that I believe because of the same reason that the Massapust lab was, he found that if you put tumors into mice that have been exposed to CMV as infants, and then you start injecting peptides to CMV, if you do that, you can get the tumors, if they grow like that fast, normally they will grow more slowly. If you do that with these viral antigens plus you add poly-IC, and you're going to say, what's poly-IC? Poly-IC is a synthetic thing that looks like double-stranded RNA, which sends a signal to the pathogen activating response to activate all these downstream pathogen, bells and whistles that make the immune system thinks there is a viral infection. If you add that plus these viral antigens, you get zero tumor growth. More excitingly, if you try to come back later and put another tumor in that mouse, it will not ever take. It will be rejected. So they can produce antigen spreading, epitope spreading, and eliminate the tumor. So I was blown away by this paper, and I was thinking about that and wondering if it had anything to, I've been studying CMV, and now CMV could be a way to bypass this tumor antigen issue. And I happened to go to a party or a dinner party with a friend who had worked as a viral immunologist. So basically, to summarize this sort of concept, what we're talking about is leveraging, putting pathogen antigens into the tumor, also putting in molecules that signal those alarm systems. If I go back to this slide, the poly-IC, what it actually did in John Schiller's study was activate toll-like receptor 3, which is ancient response that all mammals have to a viral pathogen. And that activates all these cytokines, and that brings in natural killer cells and activates T cells, and that causes them to attack the tumor. So we happen to have been working with the folks at Harvard and Boston creating a brain slice culture to test Nino Chioka's viral oncolytic virus treatment. And we have this little thing in the lab which can slice thick slices and culture them in dishes. So I said, why don't we create an ex vivo system with our own patients' tumors? And we now have this system going where we got funding for this to put tumor slices plus patients' white blood cells and then modify them with various things. In the first, sort of in the control condition, you'll see here's a GBM. We've stained it after doing this, after it was gone overnight, for CD8, which is cytotoxic T cells. And that just shows you how few cytotoxic T cells are actually in these tumors. And this is using the HDT bio mRNA that expresses a green fluorescent protein. So this is a GBM I resected, and we injected it with this molecule, let it sit overnight, and then sliced it and let it go another day. And you can see that this protein is expressed throughout the tumor. So we're thinking we have a strategy now that we could use to get stuff expressed in GBMs. And we've just started doing experiments where we co-culture the GBMs in these thick slices, and then we stain them after co-culturing with the patient's own white blood cells. And in some cases, we're giving a molecule that they make from this company that activates toll-like receptor 3. We haven't done the quantitative analysis, but it seems like we are increasing the white blood cell infiltration and mononuclear macrophage infiltration into the tumors, but we have a lot more work to do. That's preliminary, but we have a system to test multiple mechanisms now. So we have the system, and now I told you earlier that my funding for CMV got shut down pretty much about 8 years ago. So a brilliant guy in our lab said, there's a really cool concept I'd like to explore. So Dr. Karimi in the lab had read this paper, and so I'm going to sort of pivot and talk about how we pivoted and how that may all come back to help us. This is a paper written by brilliant people, including David Baltimore, the Nobel Prize-winning scientist. And the concept that most people don't know, and I didn't really know very well, I knew what splicing is. When DNA encodes for RNA, and we all know that RNA is like beads on a string, and it gets spliced. So if this protein is the normal protein in your body, this alternative splicing could encode for another protein. What I didn't really know is that during development and in cancer, often you will have alternative splicing that helps the cancer survive. It's a thing that's been known, and no one had really investigated it in GBM, although a couple papers had just done some quick and dirty RNA sequencing and said, yeah, there's some alternatively spliced genes, but so what? So this very brilliant guy in my lab said, why don't we see if we can take a holistic view? So we looked at the databases for RNA sequencing for GBM, and we found thousands of putatively alternatively spliced proteins, genes, and hundreds of those encoded for potentially for protein areas that were not in normal cells in the body. So our sort of goal for the preliminary work, and when we applied for the grant was we want to find 20, 10, 15 of these that we can see that are commonly expressed in GBM. So this is just an example of some of the preliminary data we did that led to a current funding from the NIH that we have for this project. I typically, when I take out a GBM, will take out, if it's deep, a small bit of normal tissue on the way in. So we'll have matched pairs of normal and GBM. So we went to this database and we used RNA sequencing to find putative alternatively spliced genes. And then we looked at mass spec data and we found that there were some that encoded for proteins. And then we picked a few that looked like good candidates. This thing, interestingly, is a critical gene that if it's, that prevents glioblastoma stem cells from growing. So if it's alternatively spliced, you could assume it's not functioning. So here we have tumor and normal from the same patient, tumor and normal from the same patient. And you can see that the normal protein is expressed at this level. And then in the tumor, it's up here and down here. And in like 6 out of 8 of these guys, this alternatively spliced portion is here. So our concept is this could be biologically relevant, but even more importantly for our immunotherapy, if this thing is producing chunks of protein that are not seen in the normal body, but only in glioblastoma, then we have a potential way of making new vaccines. So now we're in a deep dive on this with a group at Ohio State James Cancer Center, where we're sending them tumor and non-tumor proteins that have been cut in little sections. They do RNA-seq with long coding RNA-seq analysis and mass spec. We had a brilliant student in the lab who's now a Yale medical student who came up with an algorithm for computer analysis of these peptides. So you can run through and find peptides that are expressed highly in tumors. These 3 red things represent 3 different GBMs in the corresponding normal. So we have an algorithm for finding peptides that are actually present from mass spec in tumor and not normal. And what we're doing in this grant is we are basically, as shown in this sort of summary slide, taking normal and tumor. We're doing RNA-seq. We're finding areas that are expressed in tumor but not normal. We're determining with bioinformatics which of those could encode for proteins. Then we're doing the mass spec itself to see if they're actually present in the proteins. If we come up with protein antigen candidates, we have ways we can determine whether or not they may actually be antigenic to T cell receptors, or if you could make antibodies to these, or if you could make CAR T cells. So the ultimate goal of this portion of the project is represented here in this sort of overview slide. If we have 12 proteins, A to L, and you have 6 GBM patients, what I'm hoping is someday you can say, hey, this guy, number 5, he's got B, D, E, F, I, J, and K proteins in his tumor. And we can pull off the shelf T cells or vaccines to those. All right. So finally, I'm going to kind of try to synthesize all this and get to what we hope to do in the future, and we've started. So I've told you that I think in order to eliminate GBM, we have to take that slide that showed equilibrium and escape and push it back to the elimination phase. And I think we have to do that with a full-on frontal attack by all of these in sync with pathogen peptides, activation of toll-like receptor pathways and other viral pathogen receptor pathways, activation of damage molecules, and if we can discover new tumor-associated antigens, activation with those molecules by vaccination. And so the last thing I'll mention is a concept that's kind of out there, but it's being done, and it's based on preliminary conversations we've had. You know, if you really, really wanted to just hammer that first innate immune activation, it would be wonderful if you could have cells that actually would just get in there immediately. And so I've been working a lot with over the years with Bert Neighbors at UAB, who was one of my original collaborators. He's the head of neuro-oncology there. And he, like I, feels that we just need to be much more aggressive. At UAB, they actually have a clinical trial where they have patients who have biopsy of GBM. They place Rickham reservoirs into the tumor, center of the tumor, and then they inject gamma delta T cells that have been programmed to kill those tumor cells over time. So it's not unthinkable or crazy to consider a situation where we could take NK cells, macrophages, even modified NK cells that may have receptors for EGFR or something like that. And we've been in very, very preliminary discussions with another biotech company here in Seattle who takes cord blood, and they have these NK and myeloid cells. And so I kind of look at this as the Navy SEALs Black Ops approach. If you take a tumor and you put in NK cells immediately with all these other damage and pathogen-associated receptors, you may be able to create a massive cytokine storm that can reboot that innate immune system activation that can then lead to further activation of of the adaptive immune system that can lead to T cells coming in, attacking the pathogen, and having epitope spreading. So the algorithm that I've been talking to this biotech company here that I'm working with is that we should take a very methodical approach based on safety, but try to ramp it up at each level. So the first thing to do is vaccinate with PP65 and GB. So we're doing that now. That's a no-brainer. You can stick a vaccine into someone's muscle and get a vaccine response. What I want to start doing is doing biopsies, for instance, in recurrent GBMs, where we then inject some of these pathogen-activating molecules using the RNA, self-amplifying RNA. That will cause the tumor to send out signals for the innate immune system. Next, what we want to do, and this would be based on some of our studies in the lab using those tumor slices to prove it's actually working. Next, we want to inject either peptides or the mRNA for CMV, because there's huge evidence now based on John Schiller's lab and Masupust's lab that activating CMV, some people have 10, 20, 30% of their entire T cell repertoire in their circulating T cells is just specific to CMV. So if you can call in those troops, you'll have a massive potential immune response. So what I would like to do is put in these activators of the innate immune system plus CMV antigens, and then do this in conjunction with a peripheral vaccine. So now you're ramping up the peripheral adaptive immune system, you're activating antigens in the tumor, and you're making a holistic approach. Potentially, we could someday find these alternatively spliced proteins that we could use as an additional vaccine for the tumors. Potentially, we could inject some of these downstream, some of these adaptive immune cells like NK cells from this company that I just mentioned. All of these things would be done, I would assume, in conjunction with checkpoint inhibitors to PD-1 and CTLA-4 to get the biggest bang for your buck, because these are the molecules that are preventing the T cells in the tumor from functioning in the first place. And then one concept that I have heard from some people that are sort of thinking way ahead of the curve, what if we did this and then a day or two later did a gamma knife treatment to the tumor? That would release massive amounts of damage-activating damage-activating molecules, and that may enhance the ability for the whole process to work. So some crazy thinking over the years may have led to some unusual, unorthodox approaches, but I believe, I've spent my whole career approaching GBM with a little nitpicking, incremental tweak here and tweak there, and we've literally gotten nowhere. So I'm hoping that by focusing on a systems approach, we can create a massive immune storm, cytokine storm, call in the adaptive immune system, and eliminate the pathogens with epitope spreading at the same time to eliminate the tumor. So key observations for this talk. The CMV vaccine for GBM appears promising, but that alone I don't think is going to be the final word. The tumor microenvironment is very opaque with inhibitory molecules. Pathogen signaling with viral inoculation may overcome that inhibition. If we could co-stimulate the immune system with molecules like toll-like receptor, we could activate and further catalyze the innate and adaptive T-cell responses. We're trying to identify novel tumor-associated antigens that are associated with GBM, and hopefully we'll have off-the-shelf molecules someday that we can use as additional personalized vaccines for patients. And then the possibility of delivery of NK and macrophage cells directly to the tumor would be wonderful. So, I've been doing this a long time. We haven't gotten anywhere, and I don't think this is, as Winston Churchill said, it's not the end. It's not even the beginning of the end, but perhaps it's the end of the beginning. I think we're closing in on some things that would actually bring major changes to the way we treat GBM. And I have to thank so many people, from folks at UAB, Lily Sorociano at UCSF, my colleagues here, Parvi Hoti, who runs the lab here, Brian Searle at Ohio State, George Sun, the student at Yale, Yoon and Lee, who work in the Ivy Center lab, colleagues at HTT Bio, Dr. Bonham, our neuro-oncologist, and Nathan Hansen. And then this work has been supported over the years by SPORE grants from UAB, UCSF, R01s, R21s. And in particular, I'd like to thank the Susan McGregor Foundation, who gave money to kind of keep us alive during these last few years where there's no one wanting to support CMV research. So I want to thank everyone so much for allowing me to talk.
Thanks, Charles. That's a lot of amazing work. It's hard to sum up.
A little too much to.
Well, it's hard to sum up, years of work, and some very difficult years, I'm sure, in there, being a voice that wasn't maybe always supported. So I think that's, a testament to your perseverance. When I look at this and I think of peptic ulcer disease and what a relatively simple condition that is compared to this, and yet how much pushback and resistance in that. I mean, that whole story. So it's understandable that you would encounter as much resistance as you have. Can you first start by telling us a little bit about, again, just reiterate the trials that you're starting here and what the first patient is going to be, and just reiterate how that works particularly for people who might have patients that could be eligible.
Yeah, the VBI trial is basically a peptide, it's a viral-like particle that has the glycoprotein B and PP65 antigens. And the patients basically get standard of care therapy for recurrent GBM, plus at certain intervals, injection of this vaccine. It's pretty simple. It's like, getting the flu vaccine. So that in and of itself seems to be helping patients, and the company has correlated that with a few markers of CD4 activation, which seems to be related to improved survival, and activation of the ability of T cells to cross into tumor tissue. There's some certain blood markers that they've used. So it seems like, a lot of these things are promising, but it requires the actual functionality of your immune system to get into the tumor to get activated. But 44% tumor response rate for the phase 1 study is pretty exciting. But I think there's so much more we can do. But I'm very excited that we're actually doing a CMV vaccine trial now.
So the eligible patient is de novo GBM.
Recurrent GBM.
Recurrent GBM, sorry. And do they need biopsy for that, for the recurrence?
I think the criteria are pretty open. I would have to go back to the original slide, but I think that, basically any recurrent GBM patient with a certain KPS level is eligible. And I would have to go specifically to read the criteria, but yeah.
So for a surgeon who has a patient with a recurrent GBM that they're going to be operating on,
Yep.
they would contact you ahead of time?
They would contact VBI. It's being run primarily from Columbia University and Dana-Farber are the two main centers. Yeah, we have a lot of recurrent glioblastoma patients, so there should be no trouble filling this trial. And they can contact Lippi Bonham here, our neuro-oncologist, for any specific details.
I want to just get a couple of questions from the chat. There's, when do you think is the right time to give immunotherapy following surgery for a tumor, for a GBM?
Well, I don't know if there's a right time. I mean, any time that you can get a response, but I know that most of clinical trials are set up, especially the ones that some of the institutions require complete resection. And why is that? Because the tumors are pumping out TGF-beta, which is an immunosuppressing molecule. So it's best to get immunotherapy when your tumor is not there, or only a few cells are left, and when there's not an immunosuppressive environment. But hopefully, by this kind of full-on assault to activate innate immunity, we can overcome some of those barriers and create a firestorm in the tumor that will activate this. A lot of people ask me that are not neuro-oncology people, like, aren't you afraid of inflammation in the brain? And my response to that is, bring it on. I mean, we have ways of dealing with that. We do that all the time with steroids. But it would be wonderful to have a massive inflammatory response in a tumor.
And a follow-on question to that is how does that immunotherapy interact timing-wise with more standard of care chemotherapy and radiation?
Well, Bert Neighbors, who is my colleague at UAB, he is very forward-thinking. He's one of the top neuro-oncology thinkers in the country, and he's basically fed up with Timidar and radiation and steroids because, I mean, that is the standard of care. That's what we do. But a lot of patients don't respond well. Timidar actually causes immunosuppression. Radiation and chemo alter the biology of the tumors and make them maybe more resistant to immunotherapy. So we're talking about a future where your first treatment might be immunotherapy, and you hold off of some of these other things if possible. So it's way out of the box thinking for now, but hopefully in a few years, we can have clinical trials that show that you want to treat the tumor best at the very beginning when you can get immunity activated and when you've not altered the tumor microenvironment and the immune system with steroids and chemo.
And as you sort of think about it today, what's your hunch in terms of how many GBM patients have CMV infection? I mean, do you think what percentage do you think actually have CMV infections? Are some of these antigens that are coming about for some other reason that CMV also causes those antigens to be expressed?
That's a good question. I mean, there has been a lot of discrepancy. People can't find the genome of the virus sometimes. We did in the past. My hypothesis is that it's an extremely low level of infection under the radar, which would allow it to persist, and maybe only in a subset of cells, but that the proteins are more prevalent than the genome. I would say a high percentage are positive. I mean, when we do immunostaining, we do see antigens in most tumors. People have said, well, how can it be that a patient is seronegative for CMV and has CMV in their tumor? Turns out that very good studies have shown that some of these patients don't make antibodies anymore to CMV, and even though their T cell responses are present. So I think this is part of an immunosuppression or altered immune system as well. So I don't have all the answers, that's for sure.
No, of course not. But I mean, it's so tantalizing because you are seeing these incredible responses in some patients. We effectively know that it's more than just theory that you can cure GBM. Right? I mean,
the responses seem to correlate with the ability of the patient's own T cells to reactivate, because the patients that didn't seem to respond, if you co-culture their T cells with IL-2 in the presence of CMV antigens and they don't make cytokines, then they didn't really respond. But the patients in whom you took their T cells, you co-cultured them with CMV antigens, and you got massive interferon and IL-1 and TNF production. Those are the ones that seem to respond. So it may be that some people with GBM, their immune systems are just wiped out and they have no chance of rebounding.
And then the last one, we're over time here, but the last one I wanted to ask about is some of these CMV-associated antigens, like the PP65. Could that antigen persist long after CMV is gone and you trace CMV, the antigen somehow persists, or the antigen comes from some other source?
Yeah, so that's a good question. So PP65 antigen is very robust and it doesn't dissolve easily. And so my virology colleague said, PP65 might be produced in a stem cell and then just sort of persist in all the cells thereafter without the virus being present, which is an interesting concept. The other possibility is there is a tumor antigen called MAGE, I think MAGE A10, which is seen in melanoma, and it's exactly the same as PP65. And so there's a paper out there that if you attack PP65 with a vaccine, you can have an immune response to melanoma based on that cancer molecule. So these viruses are very adept at mimicking cellular proteins. So it could be, this could all be wrong, possibly, but we have used so many monoclonal antibodies to different antigens that it seems impossible that it could be just a one-off that there's, a cross-reactivity. But, PP65, it may have another antigen in this cell that is similar.
All right. As I said, we're past time. Thanks so much.
Thank you all.
It's amazing
Appreciate
work.
it.
A lot of
Hopefully we
years
can
of
get
hard effort.
some cures going in the next few years.
Okay. Thanks again.